Hi guys, welcome back to another video from BioBuddy, and in this video I hope you guys have watched the earlier 3 videos in which we have studied about what are molecular markers and general techniques about RFLP, RAPD. Now we will be studying about the AFLP technique, which stands for Amplified Fragment Length Polymorphism. So this is a flowchart which I've drawn, guys. So in this video what is happening is that first is the DNA isolation. We are extracting out one DNA, then we are adapting a ligation, we are ligating an adapter to it. Sorry. Then amplification of the subset of the restriction fragments takes place, and then the last step, that is the electrophoresis, takes place. Let me just have water, guys. So in this video what is happening now, I think it is the most widely used molecular marker technique, and to isolate a DNA we add restriction endonuclease to it, then fragments of the DNA is formed, and then they are amplified, and then electrophoresis takes place later. So this is the procedure that I will be telling you now, so please focus. It is a recent DNA fingerprinting technique. AFLP is a recent DNA fingerprinting technique which was developed by K-Gene in 1990. This method was based on PCR amplification of selected restriction fragments of a total digested genomic DNA. So what are the basic steps involved? First is the DNA extraction and restriction using enzymes, right? So first we are taking a high molecular weight and highly purified DNA which is required for this technique. So we need to take a very high molecular weight and highly purified DNA, and restricted fragments of the genomic DNA are then produced by using two different restriction enzymes. What are the two different restriction enzymes which are taking place? We can see on the top, on the corner, we have written MSC1, that is a 4-base restriction enzyme, and we are also taking a rare cutter, that's the 6-base restriction endonuclease, that is E-core-I, right? So the frequent cutter, now what happens to this? They generate small fragments of the DNA which amplify well. So what are these two doing? They are generating the small cuts, and they are helping us to prepare small fragments which are amplified well as compared to the 6-base long sequences. Now the first step is done, that is we have extracted the DNA. We are also using the restriction enzymes. Second step will be the ligation of the oligonucleotide adapters. Now you can see in the figure I've drawn the oligonucleotide adapters, so we have to ligate them. Now these adapters, they consist of one core sequence and one enzyme specific sequence, right? So therefore adapters are very specific for either E-core-I site or MSC1 site. Usually the restriction and the ligation takes place in a single reaction, right? So ligation of the adapters to the restriction DNA alters the restriction site. In order to present a second restriction from taking place after ligation has occurred, we are doing this process, right? So ligation में हम क्या कर रहे हैं? We are taking the adapters, we are ligating it, but they are highly specific either to the E-core-I or the MSC1 site, right? Now third and the last step I think is the amplification. Amplification हमने कर दी, right? It is to be amplified, and the selection, कैसे हम select कर रहे हैं उसको? Selection is based on the fragments achieved by the primer complementary to the adapter sequence. कौन सी होती है? अब लास्ट स्टेप जो हम लेते हैं वो क्या होता है? इलेक्ट्रोफोरेसिस. नाउ जितने भी एम्पलीफाइड फ्रेगमेंट्स होते हैं उनको हमें सेपरेट आउट करना होता है और विजुलाइज करते हैं हम जेल इलेक्ट्रोफोरेसिस से. देन आफ्टर दैट ऑटो रेडियोग्राफी और फ्लोरेसेंस टेक्निक से हम उसको देख सकते हैं. सो दिस इज डन इन केस ऑफ AFLP. I hope this video is very clear to you guys. Each and every step is clearly explained. How was the adapter ligated, and how is the DNA extracted, and the restriction enzymes, how are they used? So to be specific in this, what we need to know is that the ligation of the oligonucleotide adapters, they are very specific. They are highly specific, so they are specific either for E-core-I or MSC1 site. Usually the restriction of ligation वो एक ही single reaction में होती है, तो इसलिए यहाँ पे सेकेंड रिस्ट्रिक्शन से बचाने के लिए हमने उसको एक ही स्टेप में कर दिया. लाइगेशन हम एक ही स्टेप में कर देते हैं, ठीक है. नेक्स्ट एम्पलीफिकेशन इजीली कैन बी डन. रिस्ट्रिक्शन फ्रैगमेंट्स जितने भी होते हैं उसको हम सिलेक्ट करते हैं, वही चीज़ हम सिलेक्ट कर रहे हैं ताकि उसी को हम एम्पलीफाई कर पाएँ. एम्पलीफिकेशन के बाद जितना भी सिलेक्शन होता है इट इज़ अचीव्ड बाय यूजिंग एक प्राइमर हम ले लेते हैं. प्राइमर को हम उससे कॉम्प्लीमेंट करते हैं जो उनसे भी DNA सीक्वेंस से हमें लगता है कि वो ईजिली कॉम्प्लीमेंट हो जाएगा, ठीक है, वो करने के बाद हमारा लास्ट स्टेप क्या होता है, इलेक्ट्रोफोरेसिस. इलेक्ट्रोफोरेसिस यू ऑल्सो नो टिल नाउ. आई होप यू गाइज आर क्लियर विद द इलेक्ट्रोफोरेसिस, दैट व्हाई इज द इलेक्ट्रोफोरेसिस बीइंग डन. इलेक्ट्रोफोरेसिस इज बेसिकली बीइंग डन सो दैट इट कैन ईजिली बी विजुअलाइज्ड. हम उसको ईजिली विजुअलाइज कर पाते हैं. ठीक है, और उसके बाद ऑटो रेडियोग्राफी और फ्लोरेसेंस की वजह से हम उसको देख सकते हैं. एंड इलेक्ट्रोफोरेसिस से विजुअलाइज बाद में होता है. सबसे पहले हम उसके फ्रैगमेंट्स को सेपरेट करते हैं ऑन द बेसिस ऑफ देयर साइज. I hope you guys are liking the videos and I hope you guys have understood the concept